Alright folks, welcome back to the second episode of this whole chapter of just understanding and busting some myths about the new pandemic or the beta coronavirus 2 or the SARS-CoV-2 or the novel coronavirus or the COVID-19, whichever way you guys want to name this. The reason behind is that the more knowledgeable you are, you know about the topic, you know about what's going around you, it's going to help you to secure your family, and it is also going to help you to understand and guess what all necessary steps you are supposed to take apart from whatever the guidelines are. This second episode requires a deep, it's going to be a very deep dive, and it's going to be a deep learning about this virus, and it's going to go to the RNA level. You're also going to see it's kind of interesting and it's kind of ironic that we are doing this on a virus, but it's interesting because you're going to understand that like all computers have a binary language, which is 1 and zeros, right? On the contrary, nature has a language which is made up of essential amino acids. And today we are going to be showing you a small glimpse, not very detailed, yet a very informative kind of a video that will make you understand that coding of nature is far more supreme than human coding, to be if you want to put it that way. And that is the reason we are going to begin this episode again by screen recording all this stuff. So folks, I have written this down myself so that I can explain it to you better, because my knowledge on animation really sucks. So I can only show you the way I learned it and I know it. In this way, this is a very important episode because you are also going to understand the myth that, oh, coronavirus cannot be treated. That is a myth. It can be treated. Also, we are going to bust some myths about that, like what are the different methods of treating this. I'm not going to tell you drugs because you do not need to know that. That was the job of a physician. So no drugs out here. Because again, there will be like over the counter and all that stuff. It's not good to explain and talk about drugs in this pathway. So interesting thing is that this is a cellular level, which means that out here you're going to see that this is a cell cytoplasm, and on top is the virus entering the cell, and this is a cell membrane. The reason I'm showing it like this because I have to talk and I have to write a lot of stuff out here. So the S proteins, remember that I was talking about? The S proteins are the proteins that attach to the cell cytoplasm, or the cell membrane, and these proteins, they enable the virus to enter inside your cell. This is funny because there is, in this position 1, or in this whole position, there are actually 3 drugs, out of which some are like monoclonal antibodies and stuff like that. There are actually 3 drugs that can stop this process. So, and the reason behind that this process is like, this step is never even found out because you're not getting any symptoms at this time. There is no symptom at this time, and you do not know whether you had the virus or not. So basically this step is kind of unavoidable. And then comes where there is another way of entering this cell. So for example, this is an endosomal pathway that what we call, and then there is a non-endosomal pathway. You do not need to know, you just need to know that there are two ways a virus can enter a cell. One is with the help of the endosomes, one is without the help of the endosome. So once these viruses, they reach inside the cell cytoplasm, their main job is to, so the proteins allow this virus to enter the cell. Then once these proteins enter the cell, what they do is that they clear up, and the envelope that you see out here, this envelope, it uncoats, and then it releases the RNA material. Now the RNA material has got a 5-inch end and a 3-inch end. That's what we call it, or a 5 end and a 5' end and a 3' end, whichever way you want to name this. There is also one more stuff, that do we have drugs in this process that it can prevent the virus to uncoat? Basically, we do have a drug in this part where through the endosomal pathway part, but I think there is only one drug that is available, but that's very costly in this part of the transfer, where it comes through a non-endosomal pathway. So this is out of the picture, and the chances of it going through a non-endosomal pathway is actually very less. So yes, we do have drugs out here that can help and treat at the same time. Now, if you break a 5-inch and a 3-inch end, this is an exact picture. This is an exact depiction of uh, how the genetic codes are arranged in this picture. You will see that the 5' end and 3' end, and there is a lot of small subunits of this particular RNA material. The reason is that I'm showing you is this, that it is so we measure them according to kilobases, and you will see that there is so much of information inside this small tiny RNA. It has got information that you cannot even imagine that it is even possible. And this also brings to a this also brings to a very important conclusion, or actually brings us to a very important topic, that we are striving to go to Mars, we are striving to set up a base station in 2024 by moon and everything. But the thing is that we never did a study about each and every genetic code. Yes, there was only one study, and there's only few codes that we know about. We do not know more than that, and that too, it was done in United States, and I think other countries scientists are totally non-funded, and nobody does this research anymore. Why, I do not know. I mean, like, come on, it's 2020. This thing, or genetic coding, is the future. If we know how this genetic codes, they come together, and what are their end product is, we would understand a lot of stuff. But then again, a lot of researchers are non-funded, and that is so sad that today we are suffering the consequence of putting all of these resources that we have into more developing drugs on these processes, like just develop drugs on each and every process, rather than understanding the genetic pattern of different sort of viruses or bacteria. Well, bacteria is combatable, but viruses are very tricky, and that is the reason you will see there is no vaccines for any sort of RNA virus. Why is it so? Let's dive in and see that very quickly. Now, the 5-inch and the 3-inch end then goes through a translation process and makes these two, which is PL Pro and 3CL Pro. Do you need to remember this? All you need to remember that they go through a DNA, goes through transcription, becomes a RNA, and RNA goes through a translation and becomes a protein. Okay, now without further wasting your time, let's go and draw a couple of 5-inch to 3-inch end, because there is a lot of 3-inch and 5-inch ends out here. They're innumerous, okay? So all this genetic material, so basically you have to understand that there is a replication process that's going on. There is a translation going on, and very subnuclear level, there is a transcription going on also. So, but it's in the cell cytoplasm, it's more replication and translation. Now comes the important part of the nucleus. So, if you know a little bit of biology, you know that nucleus has got these tubular-like structures. Who can remember? They are surrounded by this. This is very important, because this is rough endoplasmic reticulum, or RER. Now, this RER's job is to take the virus. It has actually two jobs. So, the virus or the RNA comes inside and enters into the nucleus, where your DNA is. And your DNA, with the help of mRNA, actually this is going to come in, with the help of your body's own mRNA, it's going to come in, and it is entering in your nucleus. Your DNA reads what's going on with your with the whatever information has been bought or brought by this RNA. Your DNA reads it, and then your DNA, which is again essential amino acids or essential amino acids. So this is your DNA structure. So your DNA, which is again like your uh, core of your genetic coding, like that reads your RNA and imprints whatever this information has been bought. Now you might think that this is the end fate of this virus, is that's going to be unpacking. So you saw this. So the thing is that now you know that the genetic materials goes inside the DNA, right? Now you might think that this is the end. That's it. The virus is now dead. It has completely dismantled itself, and this is done. You're wrong, because in bacteria the end thing is phagocytosis, which is a way where body's innate mechanism or defense mechanism engulfs the bacteria totally, and after that the bacteria ends its life. Sometimes, most of the times, but there are other factors. I mean, I should not say that it totally ends its life, but it becomes much more weaker, because there are some bacteria that keeps on living, and it will be, like excreted out, like Escherichia coli and other stuff. But more or less, after phagocytosis, a lot of bacteria lose their power and everything. But this is not the scenario with viruses at all. And before we jump into the next step and what happens next, there are some mythbusters in this step. So mythbuster is that, can the virus be treated? Yes, as you can see, there are like all of these stress steps. There are multitude of antivirals, and the main antivirals that we are talking about right now are hepanivir and indicimavir, and all these antivirals are getting used on a regular basis right now in hospitals and everything. And I do not know why there was hydroxychloroquine suddenly coming out of the picture anyways, which was totally not done. I mean, it was so funny to watch that somebody talks about hydroxychloroquine over international press, having no clue what he's talking about. But anyways, we're not going to go into politics, okay? So let's keep this strictly scientific. Then there is another mythbuster. The mythbuster is that replication, translation, transcription. Remember this. These are three important steps that actually happens inside a cell cytoplasm, and that's how message is carried away. Now, why is this virus so dangerous? The answer to this lies in this picture, folks. This is the picture where the answer lies. On a layman's term, even if you're not going in too deep into what is going wrong and what is going right, you will understand that it is a genetic code which is not binary. So if your binary code is 1011001 and it has an information, that is only a combination that can be, like that is input and output, input and output. So this is that sort of combination. But if you have combination that is made out of such complexity, you will understand that it is next to impossible to make like a vaccine or a vaccination, and it will take a long time to make that vaccination. I should not say next to impossible, because there is a way that you can always find stuff, and it means losing hope. No, we should not lose hope, but it is very hard to make it. So also one more stuff. So thus you'll be saying like, no, I mean, like, then there is antibodies. But yeah, you have, so if antigens come in, there is antibodies that is developed, but antibodies exit your cell plasma after some time, right? So even if you're infected, can you get reinfected? Well, folks, it's a very important question, because there is a chance of reinfection, maybe not within 6 months or so, but on the next season or so, or whenever this thing comes back again or so. The reason behind RNA viruses, they always mutate, their genetic codes always get changed, and that is the reason it's different from DNA viruses. Any DNA virus, they will have a same pattern. You can only make a vaccine that has a same pattern. you cannot make a vaccine on an RNA virus, because that virus is going to bring in a message to the DNA, and it is going to keep on changing itself. So it's very hard to make. And I'm pretty sure this explanation is not at all scientific, but I mean, to convey this message to you, this is the easiest way that I can put out the message. So next step is a very important step. So what is the fate of this virus? Does it die out there? Well, folks, this is the fate of this virus. I'm going to change the color a little bit because I'm getting bored with the same color. The fate of the virus is that hold on. The fate of the virus is this, that after this process, a lot of stuff is happening in this iPad, right? So after this process, you think that the DNA has got implanted out here. Remember this. Again, let's draw a nucleus. Let's draw a nucleus out here. And we are going to draw a rough endoplasmic reticulum out here. We are going to draw a rough endoplasmic reticulum out here. So you remember that like there were three proteins that we were talking about, which was membrane, which was envelope, and spike. There is a process which is called translation. This translation is actually very interesting because it's remaking of the virus. So what happens is that this is the most scary part of this whole kind of learning, is that process of translation, budding, and assembly. I think the most important part is this process of translation, and the next process, which is budding and assembly. This is very, very, very important two steps, and this will make you understand why is it so important, why is it so scary. Because what happens is that this 5-inch end and 3-inch end, these guys, when they unpack, when the RNA virus actually unpacks, this also creates, with the process of translation, it actually makes this proteins, because again, the gold theory is that the DNA makes RNA, or gets converted into RNA, and RNA gets converted into protein, right? So these three proteins, which is M, E, and S, is actually stuck on top of the rough endoplasmic reticulum, and with the help of Golgi apparatus looks something like this, or it's a tubular apparatus also, something like this. So if you have understanding of a little bit of cell biology, you're going to know what Golgi apparatus is there. But if you do not know, it's not a big deal. All you have to understand is this, that this genetic material is getting reassembled again, and this budding and assembly occurs again, and then guess what happens? This virus gets its envelope and it gets its proteins back again. And when the budding and assembly is done, this virus exits of the cell. So basically, this virus was never lost. It came inside the host, it infected the host, or exchanged its genetic material with the host, and then it repacks itself and leaves. And all viruses, unlike bacteria, they do not require any environmental scenario to survive. They are not environmental based, so which means that these are not bacteria that needs some sort of like moisture or air to live around. It doesn't require any sort of oxygen at all. So basically, it is kind of unbeatable kind of a scenario. So in this episode, and a very big myth busting episode of in this episode, actually it's a very big I think chapter in this that we learned in this episode, is that if we look into this, that attachment and entry, there is multiple drugs that is available for you for your treatment, and This is how a genetic code looks like inside a coronavirus. So I took this picture from a research material, and you're going to see that the SARS-CoV, so this, the first one is of the SARS coronavirus. This next is the SARS coronavirus also. Then there's the other one is a MERS coronavirus, and these are all subunits, and you will see that how they're arranged, and it is such a big information that is just happening within like few days or so. So another mythbuster is this, that so why is this virus so dangerous? This virus is so dangerous because after all this thing has happened and it has, your DNA has got the information, it takes time for the DNA to send that information to your body, and your body will take some time to react to it, because all the other cells that is having that is, you know, that you have, that is your natural killer cells and other T lymphocyte cells and all the other cells that makes up your defense mechanism will try to combat it. So the thing is that nobody gets caught in this stage. So a big mythbuster is that nobody gets caught in this stage at all. Everybody gets caught at that stage when the virus has more or less exited out of your body. You are left alone with some antigens, and now you're getting the symptoms. So you are in a stage where you are expressing symptoms that can be cough, that can be bilateral lung infiltrates, which means that your chest picture shows multiple infiltrates all around your stuff. All your nervous system got infected, and you are suffering from some sort of comorbid diseases, and hence comes episode number 4 that you need to watch, because lungs is an organ that cleans your blood and puts more oxygen in your blood, to be in a very small or narrative, or a very, you know, like a small way to understand is that what it does, it takes deoxyhemoglobin, it inputs oxygen into your thing, and that's how your system is having more oxygen, right? If that gets compensated, that means your whole system is messed around. Well, folks, again, thank you for watching this episode. I know that it stretched out a little bit more. This episode and all these diagrams will make you understand and beat all the questions that is thrown in your mind with different sort of materials that you're collecting from any sort of place, like from media, or you just learned it from your friends. Remember this, that RNA virus is a very huge, you know, like a topic to understand for a lot of virologists, and underfunded, you know, medical research is a huge problem. So, you know, like if ever you get a chance to support any kind of medical research, do extend your support for them. And also, you know, like I think new reforms should be thought about, you know, funding medical research more. And if you have any other questions, you can always write down in you know, like in the description below. I will try to answer if I get time, because most of the time I'm super busy right now with all the stuff that's going around, and I will try to answer as fast as possible. Do not put questions like what all drugs should I use or anything. Nope. This is not a channel that's going to tell you that you need to take this drug and that one, you know, like that, but this dosage and that dosage. For that, you will consult your physician. You are going to follow CDC guidelines and your local guidelines, but you take these steps on top of it. Scenario, will the second wave be bigger or not? Will the second wave infect more people? I have got a shocking truth for you, which I'm going to share with you on the later episode. But the next episode will be again a little bit of tech stuff, so that it doesn't get boring with health topics again. And then on another episode, probably next week or within a week or two, we are going to talk about how big is going to be the second wave, what is the target population that it's going to infect, and what will be the symptoms, and how the virus got mutated, and what are the new clinical pictures for this virus. So take care. See you guys on another episode. Till then, stay safe. Knowledge is only there to empower you. You should not be afraid of life. Never lose hope on life. This is not the end of the world. We are going to go through all of this. It's just to understand, you know, how better we can protect ourselves, and that's pretty much it. So see you guys on another episode, and stay safe, stay together, and you know, take care of your family. Goodbye.